Speakers came from the island, and both made it here. So our first speaker is going to be Kristi Miller, who's going to, I'll introduce in just a moment, and our second speaker is Helen Bernice Smith. So we're delighted that they're both here, and I'll formally introduce our moderator for this session, Brian Riddle. But I'll just say, Brian Riddle is the president and CEO of the Pacific Salmon Foundation, and I don't think we can have a better expert in here to moderate this session. And it's just kind of serendipitous that Brian is a geneticist by training and has been in the salmon world for a long time. The foundation, I'll let him tell you more about it, but it looks after the wild salmon stocks. And it's really quite interesting that Kristi used to report to Brian in her capacity when she first started at the DFO. So with that, just as an overall introduction, let me just introduce Kristi Miller, who is going to tell us about the FishMan Omics project, Genomic Tools for Fisheries Management. And Kristi's the head of the lab, of the Genomics lab at the DFO. She's been doing, she's had her much more than 15 minutes of fame recently. It's been kind of going on and on with the Cone Commission. And I'm sure she'll be telling us a little bit about that. So anyway, Kristi, thank you very much for joining us, and please join me in welcoming Kristi Miller.
Thanks, Brian. Okay, today I'm going to give you a brief overview of an AGIT project that we have been doing. We're in the fourth and final year of this project. And my new title is really Recognizing Dead Fish Swimming, because ultimately that is the goal of what we're doing with genomics, and I'm hoping by the end of the talk that you understand what I mean by that. The project is a collaboration between UBC and DFO, and my key collaborators are listed here. So BC salmon have been in the news a lot. As of late, it would be very hard to miss the news about BC salmon, but really even over the last decade, there's been a lot of reports in the news and on the internet about salmon, and that is because of the cultural and economic importance of salmon to the province of British Columbia. I just did a Google search over the past decade, looking at how many hits to British Columbia salmon one could find on the internet. And even back in 2001, there was almost 500,000 hits to British Columbia salmon. If you go all the way up to 2011, we're at over 15 million hits this year. And this is largely because of the Cohen inquiry, which I'll briefly explain to you. And the unfortunate thing is that one of the reasons that salmon are in the news as much as they are is because there has been a lot of difficulties in their management and generally a precipitous decline in many stocks. So if you look at the kinds of things that are highlighted in each various year, we see in 2001 high losses of Fraser sockeye, and then we see sockeye fisheries curtailed and worries over disease, and sockeye disappear in the river, and they show up late, and on and on and on. So these are the kinds of things that are being discussed in the media and on the internet, and these are the kinds of things that both managers and the stakeholders are dealing with on a yearly basis. So the reason for this is one of the key reasons is that salmon productivity for a lot of stocks and multiple species are in decline, especially in southern British Columbia. And this shows, I show on the top 2 Fraser sockeye stocks, and you can see that since the early 1990s, there's been a fairly precipitous decline in productivity in both of those stocks. But along with this general decline, there's also a lot of annual variation. So it's not a real. I mean, you have some years where there's relatively good returns, and other years where they're very poor. But it generally it's in a declining state. The bottom 2 graphs in coho show basically ocean survival of coho, which you can see over the last over 25 years has been declining. This could also be shown in Chinook stocks as well. There's no place where this has been more unprecedented than 2 years that occurred very recently. And these 2 years are just after our Genomic project started. And those was in 2009, where we had very record low returns, and in 2010, where we had unprecedented high returns. And in both of those years, importantly, management models predicted 10 million fish, 10 million sockeye salmon would return to the Fraser River to spawn. In 2009, it was the lowest returns in 60 years, with only 1.4 million fish actually showing up. This was estimated at a marine survival of about 0.4%. In 2010, it was the largest return in 97 years, with over 35 million fish showing up. And a marine survival estimated about 12%. So this is a very large standard deviation around an estimated return. And it really, this kind of thing is what motivated the development of our genomics program in trying to develop tools using genomics that might help with evidence-based management and bring in information about the condition and fitness of fish that had not been being considered in any kind of management context. Because of the very poor returns in 2009, and largely because of the lack of prediction of those returns, the Prime Minister of Canada declared a Cohen Commission of Inquiry in 2009, tasked with investigating and reporting on the decline of sockeye salmon in Fraser River and making recommendations for improved management. So there were basically two angles that were being tackled in this commission. One was sort of the scientific issues. What are the reasons for the decline? What are the scientific explanations? And what are the scientific explanations for the poor returns in 2009? And then also, what is DFO, what are the management agencies doing, and why are they unable to predict this? And how is science and policy, what's the interplay between that, and maybe that can be developed better? So this has been ongoing for a couple of years now, and this past year has been the time when there has been a lot of scientific debate. And the scientific community, we won't hear about a report about this until June of next year, but there's a number of key hypotheses that have been put forward to potentially explain the declines in productivity. And I'm only going to touch on a few of them here, two of them being fairly key hypotheses that have been debated a lot in the Cohen Commission. One is the role of disease, and another is the role of the early marine environment. Many in the scientific community have come to a common conclusion that when salmon move from freshwater to the early marine environment, this might be a period of time where there's very high and unpredictable levels of mortality. And so there's been a lot of debate about that, and I will touch on that. A third, which is not given quite as much weight, is the role of harmful algal blooms, but I do mention it in my talk, and I thought I'd put it here. I think that there's a general consensus from most in the scientific community that there's not going to be a single factor that can explain all the declines and all of the variations in various years. But what we're looking for are what are the key factors that we should be focusing more attention and research to, and how, once we can understand what factors are involved, how can we use those to predict or possibly mitigate the decline. So the genomics that we use in this program is functional genomics, so we're basically using the transcriptional profiles as a way to look at salmon condition, as a way to look at genome-wide scan of physiology. And so we've used both cDNA microarrays and oligo microarrays, both coming out of the seagrass program by Ben Koop and Willie Davidson. And so these microarrays have ranged from 16,000 to 44,000 salmon genes. And we use these to identify a number of different conditional states. For instance, whether fish are likely to be food deprived or starving, consuming different qualities of prey, under stress, and a variety of different kinds of stress, growing fast or slow, and on and on. Ultimately, what we want to do is identify signatures that are indicative of poor survival, and I will give you one example of that in my talk today. This project really involves the use of fairly large-scale microarray experiments, and we look at a variety of supervised variables. Some of these supervised variables are important in understanding differences in productivity. For instance, we see different levels of survivorship in different years. We see different levels of productivity between different stocks, between different life history types, between hatchery and wild fish, and on. And so this just represents heat maps that show some of the genes that differentiate these various variables. And I'm only going to talk really today about looking at annual differences in physiology, and also some work that we've done on disease and also fate. So we've done a lot of focus on annual variation, especially in the last year of our project, where we have a lot of samples for multiple years now of smolts, sockeye smolts, leaving the Fraser River and into the ocean. And basically, our hypothesis is if productivity is determined principally by the conditions in the early marine environment, which is something that the scientific community has been pondering over the last couple of years, then physiological divergence among years should be most apparent in the marine environment. So if the marine environment is the driver of these variations, this is where we should see the biggest variation in the fish. So we can simply ask that question with our microarray data and contrast different tissues from different species in different years. And this just shows a cohort experiment where we looked at brain tissue on the top two graphs and liver tissue on the bottom, and looked at the divergence between fish in the freshwater and the saltwater environment. These are fish from the same sample from the same stocks in the same locations, the same time of year in each year. And what we find is that, in fact, the level of divergence in freshwater, and this is only 2 years here, is just as high as what we're observing in saltwater. And if you look, I show on the bottom some of the functional analysis of those data. So, for instance, in the brain in freshwater, we see differential activation of pathways involved in metabolism and growth factor. And in the saltwater environment, we see really differential stimulation of pathways that are involved in the external environment. So, immune pathways and response to wounding and external stimuli. And in the liver, we see a lot of immune stimulation in freshwater, and in saltwater, we see a lot of metabolic shifts. This is when they start undergoing their rapid growth phases. Most importantly, in 2009 is the year where we had very poor returns. Those fish would have gone out into the ocean in 2007. And in 2008 was the year that the fish that returned in huge abundances in 2010 would have come out into the ocean. So this particular study in sockeye salmon contrasted fish between 2007 and 2010, sampled in the freshwater and the saltwater environment, asking again the same question. Among those different years in those two different environments, where do we see the biggest divergence? And really interestingly enough, the highest level of divergence, the outlier among all of those years, was 2007 fish sampled in the freshwater environment, not the saltwater environment. And so, it's really tempting to ask, could this signature be a key to the poor productivity of the smolts in the ocean in 2007? I'm not going to show you the answer to that because we haven't finished analyzing those data, but we have similar sorts of data stemming from liver and muscle tissue as well. And some of the things that we see that differentiate the 2007 smolts, which did very poorly, and the 2008 smolts, which did very well, are that 2007 fish carried enhanced immunosuppression, especially of the humoral immunity, enhanced stress, hypoxia, signals of hypoxia, and we ended up asking the question, where would that hypoxia come from? And we amplified harmful algal blooms from the gills of those fish. So there may be some merit to the hypothesis that harmful algal blooms could have a negative impact on wild fish. We saw that they had a slower growth-related genomic profile in muscle tissue, and they carried a higher proportion of fish that had this mortality-related signature, which is what the topic of my next slide is. And so none of these things are a very good formula for survival for those fish that went out in the ocean in 2007. So I'm going to just briefly switch gears and talk about a study that was done on fish in 2006, where we combined radio tracking and non-destructive biopsy sampling and genomics to try to understand whether or not salmon, whether or not we could predict before the mortality events were occurring in the Fraser River. And this is on, I'm sorry, I should have shifted gears. I'm now talking about adult salmon. So one of the other issues that have also occurred in the last decade, or actually about 15 years now, is that Fraser River sockeye salmon have been undergoing unprecedented levels of mortality in the river when they return to spawn. So they at 4 years, when they come back into the river, typically they would enter the river and about 15% of them would perish before they would spawn. In the last 15 years, the